I hope you're having a good day. This is Dr. Nolan. Once again, we're beginning to have another discussion on a completely different kind of a material. This is more of a discussion is going to be reviewing how I use Mindjet maps to help me understand material and help me organize things so I can actually incorporate it either into projects or incorporate them into my understanding of the subject matter. Now, what I found over the last few years is that sometimes complex material is becomes difficult for students to understand because they don't understand the actual starting point in which the material is actually um, sort of incorporated in. And what I'm trying to tell you is basically you need to know the major headings in any kind of material to really understand where that material lies overall in the actual discussion. Oftentimes when we begin to discuss a lot of detail, we get lost in the detail. We don't realize the overall function of what we're actually trying to discuss. So one of the things that I find to be very useful is developing what's called Mindjet maps. Uh, and Mindjet maps I actually use for almost all activities in my life, including personal, such as stock market, or things that I use for schooling, teaching students new material and developing PowerPoints. All this usually originates right here using the Mindjet maps first. Once the Mindjet maps are processed, you can actually then convert them into PowerPoint slides, as well as you can transfer them over to Word for outline formats as well. So these Mindjet maps can be useful in a lot of ways. Now, one of the things that I want you to know is that the school has developed a portal which you can access, in which you can use the academic discounts to actually purchase the Mindjet Map 8. Mindjet Map 8 runs like $150 with the academic discount. Without the academic discount, I think it's like about $300, so it's pretty expensive. So with the academic discount, definitely make sure you use our portal. The only thing you need to get the discount is to send them via fax, or you just send them via email copy of your admissions letter. So that will actually work for that. Uh, Now, I expect the Mindjet maps to be used in the sense of helping you build your knowledge base and how to actually produce these maps, but also it's also set up to help you organize your thoughts, which I think is also a very important part of the DNAP program. The way I see the DNP program is that eventually that you're going to be the next set of professors involved in teaching other CRNAs, or you're doing this as well to really advance your knowledge tremendously. I think part of the things of developing the DNAP program is that you develop tools so you can really advance your knowledge quickly. And I think this is one of those tools that if you never have been exposed to it, I think it's very important. And I think it'll be very helpful, and you'll note that by the end of this semester. In addition to that, Mindjet Map will be used for the next several classes that I hold, which I think will tell you how important I think this is. Now, what I'm going to go first with you to sort of review and show you very briefly how I use the Mindjet maps as an example is by taking one of the articles that I've asked you to read. I'm not going to go over the whole article, but I'm just going to go over the major topic portions so you can see how I begin to use Mindjet maps to help me not only learn, but also to help me teach sometimes different students. The major heading in our article was vascular smooth muscle and general anesthesia. So as you can see, that's in the major blue box here. Okay, and then it's basically broken down in the article into three minor subtopics. One is the extrinsic vascular control, intrinsic vascular control, and how the actual general anesthetics affect the vascular smooth muscle. Okay, when we look at the extrinsic vascular control mechanisms, there's two major forms of extrinsic vascular control mechanisms, which are the circulating hormones, which we're going to discuss later on next semester, and the other system is the neurovascular control centers. The neurovascular control centers, of course, has to have information that comes in to it for it to actually process prior to actually receiving that information. The afferents usually are either via baroreceptor reflexes or may also be occur due to mechanical reflexes or chemical receptor reflexes. That information arrives to the central nervous system where it is then sort of modulated and decided on as to what to do with that information, and eventually it gets sent out to the effector systems, which end up causing an effect. Okay, the effector systems, in the sense of vascular smooth muscle, is only the sympathetic outflow chain. Now you ask, why not parasympathetics? Well, the parasympathetics are really not responsible for smooth muscle, vascular smooth muscle control. It's only the sympathetic outflow. Now, one of the things that I want you to note is you can see on how this works with the with Mindjet maps is that you can actually incorporate hyperlinks into your maps, and hyperlinks are very useful if you're doing a study, or if you're visiting a website that you find very interesting, or you can find a picture in a website that you find interesting and you want to go back and review it. So in this case, for sympathetic outflow, I want to see a picture, what it would show that. You can see it blows up very quickly. It immediately links to that site and shows me the actual picture that I want to look at, which is down here, which demonstrates a sympathetic nerve fiber sending out its neurotransmitters, which then affect the vascular smooth muscle. Okay. In addition to that, the sympathetic outflow affects basically four portions of the cardiovascular system. It affects resistance vessels, specifically the arterioles. It also affects the capacitance vessels, which is your venous return. So it vasoconstricts the veins and increases preload return. It increases myocardial activity by causing direct stimulation of the myocardium, and causes increases in heart rate by alteration in the SA and the AV node conduction velocity. In addition to that, that's pretty much all the sympathetic output. Okay. Now we're going back to a discussion of the intrinsic vascular control mechanisms. Something that the article refers to very briefly is the myogenic mechanisms, which is the actual mechanisms that are the muscle tone itself is important for actually intrinsic vascular control. It doesn't go into a lot of detail on that. Instead, it goes more into the detail of vascular endothelium control. The metabolic metabolisms are also important, which is not something that's really directly discussed in this chapter. The vascular endothelium, though, seems to respond to neurohumoral stimuli as well as physiochemical stimuli. What we see with this situation is it causes release of vasoactive mediators, okay, which can either vasoconstrict or vasodilate the actual vascular smooth muscle. One of the things that the article discusses into a lot of detail is how the actually direct role of calcium is, specifically with discussion on the role of calcium, where it's located at. The calcium is usually found in the intracellular stores, such as cytoplasmic reticulum, or as extracellular space, which is the actual cytosolic free calcium. Okay, when you have an increase in cytosolic free calcium, which you'll see is vasoconstriction, you may also see calcium begins to bind to I think what's called camidulin. Okay, and what this nice picture shows you here is part of the article. Okay, so which I've actually cut out and incorporated into my my study, so I can actually see how this actual mechanism works. This black line here would indicate the plasma membrane with the receptor location, and the agonist, in this case norepinephrine being the major agonist, would bind to the receptor, causing a G protein activation, leading to the release of these different cascades that take place. And specifically, they're talking about during this discussion is how calcium, when it gets freed up, which is right here, binds to calmodulin, and calmodulin then will eventually bind to calcium and calmodulin bound together will eventually affect and convert the myosin light chain kinase to convert myosin light chain 20 to a phosphorylated myosin light chain 20, eventually causing a binding with actin and causing contraction to take place. Now, as for decrease in cytosolic free calcium, you will get, the end product will be a vasodilation. Sorry, let me go back. Vasodilation. Okay. You will also see an extrusion of calcium out of the cell, or the uptake of calcium into the sarcoplasmic reticulum will actually cause a decrease in calcium and as a consequence cause vasodilation. In addition to this, you have signaling pathways that are available. These signaling pathways are things like the myosin light chain kinase, which I was talking about before. The myosin light chain kinase is an enzyme responsible for the phosphorylation of myosin light chain 20. Okay, and when it does that, when it phosphorylates it, it causes the formation of actin, and eventually the binding to actin causing contraction to take place. There's other things that can actually modulate the sensitivity of MLC20 phosphorylation. And the other thing that is discussed in this article is how calcium calmodulin dependent 14 kinase 2 is also extremely important as a secondary pathway. Now, there's also other roles that are taking place in this discussion. And the role of phosphatidylinositol cascade, which is activated, okay, leading to vasoconstrictive agonist effect. Things like norepinephrine, angiotensin 2, and endothelin 1 are typical examples of this kind of cascade event. And then with the receptor activation, it leads to the activation of hetero trimeric GTP protein binding and activation of phospholipase C, which then hydrolyzes phosphatidylinositol diphosphate, and as a consequence breaks it down into inositol triphosphate and DAG, or what's called deacylglycerol. The inositol triphosphate stimulates sarcoplasmic reticulum release of calcium, and this eventually leads to IP3-induced calcium release as well, which is calcium-induced calcium release will open a channel, which then as a consequence helps with increasing intracytosolic calcium, which increases contraction. What I basically described to you briefly is basically this pathway here. Here's where the agonist binds to the G protein. The G protein then converts phospholipase C, which converts inositol biphosphate into inositol triphosphate and DAG. Okay, the inositol triphosphate binds to the type 2 calcium releaser, which then releases calcium from the sarcoplasmic reticulum, as well as the calcium then further activates the CICR, okay, which is, you go back here, you can see it, what I'm talking about, okay. Make sure I'm just showing you here. Make sure you can see it right here. Okay, there's a inositol triphosphate induced calcium release, and then you have the calcium induced calcium releasers, which is the CICR. Okay, so you can see very quickly how this helps you visually format your thinking process, so you can able to describe a very complex reaction that's taking place in this article. Now, in addition to that, I want you to understand that all these things can then be converted into different kinds of formats. So I'm going to go over just briefly to show you another pathway here. General anesthesia, I've gone over all these different factors in the article, okay, and it's something we'll discuss in one of our discussion periods. But you see, you can actually break it down into all these different pathways that help you study. Now, I'm going to stop this presentation here, and I'm going to show you how we can actually take some of this information and then begin to use it in other formats. Okay, so this is all the information put together. Okay, you can see there's a lot of detail. So I'm going to show all the levels. Okay, let's do the same here with all the levels. I'm sorry. All levels. Okay. Okay. Now, what you can do with this is you can actually take this and you can do an outline view. Okay, so it actually shows you all the different topics based on outlines. It can give you a multi-map view, which is when you have multiple maps put together, you can begin to link them if you need to. Okay, so this software gives you a lot of ability. You can send it as emails, which is what I want you to do. Is you send this thing to me as an email, and then I will look it over. Um, okay. Okay, going back to map view. In addition to that, you can actually convert it. Okay, going to here, you can begin to save it or export it. Okay, you can actually say export it to a Microsoft PowerPoint slide. So I'm going to show you how this works. I'm going to format and export to Microsoft PowerPoint. Show you what it does again for me. Okay, sometimes it does stupid things like that. Sorry, it's taking a little bit of time, but sort of want you to see how powerful this is. Okay, open the folder now. Okay. Okay, so now it shows me pretty much all the PowerPoint presentation, and it begins to help you develop your slides. Of course, it needs a little bit of work on, but it gives you a general idea how I use it to help me develop slides and different things. Okay, and of course, it shows you how the structure of the PowerPoint is here. How it's all broken down for you. So it's not like you're having to do the work 3 times. You just do it 1 time, and it sort of assists you in making your PowerPoint presentation. Okay. In addition to that, you can actually convert some of this material as well. Okay, you can export it to different things like Microsoft Word. Some people like using Visual also, which converts this into another format. And you can also export your task information to Microsoft Outlook if you want to have certain tasks that can be incorporated. So there's a lot of things that this software can do for you and to help you really establish your thought processes very quickly. Now the other thing I want to show you that it does that I find useful as well is that you can do either attachments. Okay, you can attach files, or you can actually put on notes. So on metabolic mechanisms, you can actually write a whole thing on notes here, which makes it it's not incorporated into your major heading, but actually just discusses metabolic mechanisms. Like I can assign something like look this up later. So it gives me an ability to review it if it indicates me to do that. Okay, and then I can actually hyperlink something like this. I can look for the hyperlink. Okay, let's say I'm going to look for an issue where it talks about metabolic smooth muscle. Okay, and I can look it up. You can see actually put a journal article if I want to on this. So I'm going to link this. I'm going to just accept this, copy. Okay, I'm good. Put it back here. Okay, I'm going to say OK. So later on, if I want to come back to this, I can come back to metabolic mechanisms, and I can pop open this article, and it immediately opens it up for me. See, this is how I think this is a very powerful tool in the sense that when you're trying to do a lot of different kind of research and you're trying to look up a lot of different sets of information, it becomes very useful in being able to add to your already known knowledge base. It's also a very easy way to study for exams because what you can also do for examinations is you can take and basically make it into a major heading and then say, okay, what are the vascular smooth muscle effects due to general anesthetics? Okay, and you can pop it open one at a time. It says what affects the cardiac function, affects the cardiovascular reflexes. There's also direct action of general anesthesia on vascular smooth muscle. How does that occur? And you can see it's due to vascular reactivity effects, and it could be to vasoconstrictor response. Okay, and you have vasodilator response. The volatile anesthetics vasoconstrict, they inhibit contractile response to potassium chloride or norepinephrine. It can cause vasodilation, and the IV anesthetics unknown effect on the vasoconstrictor response. So you can see how you can begin to develop your mind maps to actually help you study for exams as well. Okay. In addition to that, if you feel like it, if you in some situations you can add sketches, okay, which is you can actually create a sketch, and especially if you're using a tablet PC. So there's a lot of different things you can actually do. Okay, I hope this is helpful to you and sort of giving you an idea of where and how I want you to develop your mind maps. Okay, the way I'm going to grade you guys for your Mindjet maps is how useful they are when I look them over, and how useful they are to you. As you get further along in the course, your Mindjet map should become more complicated and should be more versatile once you learn the software a little bit better. So initially, I just want you to get the basics, get the major headings down, make sure that you have hierarchy-based information, and that you put enough detail in there that it sort of helps you understand the material better. Okay. Well, I hope this has been a helpful, brief discussion. Hope you have a good evening. Thank you.